suppressed. It's not as dramatic as we see in the thermally sensitive one. And so even though these genes are temporally disconnected from when we actually see bleaching, we think that these may be actually predictive of how much bleaching is going to be seen in our strain based on the fold change expression at these early time points. And so if we kind of plot these, in red will be the average of the thermally sensitive strain and the apo strain, and here's the more thermally tolerant endo. And we see that we do get this upregulation at this early time point, but again, it's to a lesser extent. And based on that lesser extent, we see a level of bleaching that is not quite so much. And so first we're asking, are these genes causing bleaching? We really don't think so because of that temporal disconnect. But maybe these are either protective or there's something going on to be predictive of our bleaching. So a conceptual model that I have right now is that in our more thermally sensitive strain, they don't have this ability, or these are not protective, and we get more bleaching. Potentially, these algal symbionts in this endo strain, those that are more co-evolved together and can deal with these stressors, either the algae are doing something to block gene expression, and so you don't get much of a response from the host, and thus you get less bleaching, or maybe the algae are actually doing something with their own gene expression to combat some of this stress response. And because of this interaction, we don't see as much bleaching. So from here, we do see this, what we're thinking is a generalized stress response to heat stress. So we've got this pattern of genes that are up early and then come back to baseline. We see this similar response in both the apo and sym conditions, which are making us think that it's really not dependent on the presence of the symbiodinium. And then we've got these genes that are upregulated early on and stay up. But again, now we don't know what these genes are. We have to go back in and clone them individually and see what they might be doing. Are they involved in bleaching? We don't know. And then we also see this heat stress response is suppressed in this more thermally tolerant. And so either these genes seem to be somewhat predictive, but now the question is, what are the algae actually contributing to this? So we haven't looked at any of the algal gene expression yet, but we've got those sequences in our database, so we can go back and see now what are the algae doing in response to this thermal stress, and maybe are they expressing a similar suite of genes that might be leading to this overall dynamic interaction. So I'll end there, just thanking Team Anemone. So this is the Pringle Lab back at Stanford. Helped a lot of this work, especially Philip Clevis. There's John with his little Aptasia shirt, and we all made hats for the meeting. And of course, not only are those people involved, but over the span of, you accumulate a lot of collaborators, great colleagues and friends, some of which are in the audience here. Thank you again for the invitation. It's a pleasure to be here. Thank you. Time for questions before everybody gets a beer? I'm going to drink a beer. Yeah, please.
What's the source of Serratia margens in nature?
This particular strain, it's hypothesized that it was mismanagement of sewage runoff in the Florida Keys. So back when they were still on septic systems, there was a lot of misregulation, and a lot of that got flushed in. And so the thought is in the mid to late 90s, mismanagement of sewage got into the Florida reef track, and Aaron Lipp and colleagues at University of Georgia did a lot of that source tracking work to bring it back.
But it's normally a bright red bacteria. Right?
Yeah, so many of them are pigmented. This one is not, right? So it doesn't produce that progesterone pigment. Yeah. So it's a question that is this one actually a human pathogen, or if it's another strain? That's kind of the, and so I've always kind of answered that question the way I just did, that somebody else did the source tracking, and that's what they tell me. But I don't know, and I always had to convince EH&S that was this really a human pathogen, because I had to treat it as such and use it at BSL-2 levels, even though we have no real indication that it actually is a human pathogen to the same extent that other strains are. Yeah.
I know this is a sensitive subject, but in terms of your clade A versus clade B, what's the difference into genetically that it would respond so differently with the same background?
That's a really good question. We don't have much genetic information on the clade A. The genomes are huge. There have been a few published genomes now of varying qualities. I don't really know a lot physiologically. The big debate in the coral field is clade C versus clade D. And so Aiptasia don't normally associate with clade A or C and D. We now have them associating with clade C and D. And so we might be able to ask some of those questions. But again, because it's a novel association, I don't know how representative the Aiptasia system will be. But we see a similar story with clade C and clade D in that clade C is more thermally tolerant on average if we go across the entire clade. But they seem to be maybe a little bit better symbiont. So it's less kind of energetic cost for the host to harbor clade C, and they're more photosynthetically productive. But then if a thermal stress comes about, they'll bleach faster. Whereas a coral that has clade D don't get as much photosynthate. Clade D isn't as productive. It requires more energy to maintain it. But now if a thermal stress event comes in and you harboring clade D, you may bleach less. So it kind of becomes a trade-off, and often corals that are harboring clade C are harboring a mixed population, so they've got some D, some C, and then what you'll see is a bleaching of the clade C, retention of the clade D, and that's what repopulates. So it could be, but again, what about them is making them, different levels of the ability to be a good symbiont? I really don't know at a genetic level. Yeah, Lauren. Hi.
Do you see being able to perhaps seed thermal tolerant symbiotic in a bleached coral? Do you think that's a potential endgame?
People are thinking about it, and yeah, maybe. This is something that kind of came up at the end of our work with the potential of the probiotic bacteria, right? What's the potential of actually applying that to a reef? And obviously you can't go out and spray a reef with either beneficial bacteria or potentially beneficial symbiodinium, right? I don't think anybody's going to allow that. But in terms of nurseries and reef aquaculture, if you can kind of seed them and get them established in individual colonies in your nursery or in your aquaculture and then use that for transplantation, maybe. We don't really have a good sense, especially at the bacterial level, how stable these kind of modified bacterial communities might be. If we can get them to establish in the lab, will they remain once we put them out into the environment? And so that's one question that I'm kind of asking with the Aiptasia, is I want to take some of our Aiptasia that we've been culturing in the lab for the last 10 years, looking at how their microbial community looks compared to those that we collect naturally. And then if we bring those natural ones back into the lab, how do they change? But yeah, I know, right now there's a lot of effort to identify genotypes of coral and symbiodinium that are the more resilient strategies, and that's what people are focusing on for restoration efforts. And so I don't know how much of that is more targeting those combinations that exist naturally and then trying to asexually propagate them in your nursery and then transplant those back out, versus kind of bioengineering, taking the best coral genotype and the best symbiont genotype, mixing them together, and then either put them back out or, even seed, a reef with beneficial symbiodinium. I mean, it's kind of happening in nature. So we've now got influx of clade D symbiodinium into the Caribbean that normally were not there. So from ballast water and other things, clade D now has made its way in from the Indo-Pacific and are now starting to get in species that normally didn't harbor clade D. And so what kind of ecological consequence of that may be, who knows? So it is kind of happening, but should we be the ones targeting it? I don't know yet. Yeah, that's the question for managers. So it's found all across the world. The strain from Florida is right now designated as Aiptasia pallida, and that strain that harbors the clade A is really only found in Florida and some parts of the Caribbean. And then our H2 variant, the Hawaiian strain that has the clade B, is sort of the more pandemic strain. And so if you go anywhere in the Indo-Pacific or Hawaii, you'll find Aiptasia normally harboring these clade C symbiodinium. People that are really keen on taxonomy with Aiptasia now are arguing that these two, so the Hawaiian strain is labeled as Aiptasia pochella. So right now labeled as different species. we can get them to hybridize in the lab and form viable larvae. We can't get these larvae to settle out and grow into an adult polyp yet, so we don't know what the F1s will really look like. But now people are arguing that Aiptasia pallida from Florida should be a genus called X-Aiptasia. And some people have adopted that nomenclature. I have not, because I don't care. Because in the lab, again, we can get them to make larvae, and it's one thing to have two different species make viable larvae. I don't think there's ever been an example of two different genera forming viable offspring. So right now, the Aiptasia pochella is the more global pandemic strain that you can go everywhere and collect it. You don't really see it too much on reefs, but if you see it on dock pilings and mangrove forests, you'll see them on the roots in kind of clumps or aggregates, kind of in a more shaded environment, or in your local seawater aquarium, to the displeasure of whoever's taking care of it, probably. So it's got its start as an aquarium pest. And so if you're an aquarium hobbyist, if you get live rock, you'll have an Aiptasia that comes in. They grow faster than your coral. They grow in high numbers. There's a commercial product called Aiptasia X, which is basically injecting cement into the anemone, and it solidifies and kills it. So what's not good for the aquaria are really good for me. Yeah? That's right. That's right. Yeah. And so we're very careful not to have those in the lab. All right.
Okay, let's thank Cory. Go ahead.
Thanks again.